Co-binding of chromosomes. Co-binding of chromosomes is another method by which we stain the chromosomes. In this method, we use a dye. The name of the dye is quinacrine. In case of G-banding, we use the dye that was Giemsa, but in case of Q-banding, we use the dye. The name of the dye is quinacrine. So as a result of this type of the banding that is almost similar with that type of the banding which we have seen by using the Giemsa stain. So Q banding and the G banding, they have similar patterns of the dark bands and light bands. This is the procedure how we stain the chromosomes while performing the Q banding of the chromosomes. The slides, they are dehydrated by dipping in alcohol with decreasing concentration of first with 90%, then with 70%, then with 50%. So slides, they are dehydrated. Once slides, they are prepared, then they are dehydrated by using the alcohol, and the percentage of the alcohol is in a decreasing pattern. First we use 90% alcohol, then 70%, and then 50%. 1 minute for each of the procedure. Once we have dehydrated the slides with the alcohol, then we rinse the slides with distilled water. Wash the slides with phosphate buffer. After the washing of the slides with distilled water, slides are washed with phosphate buffer. Then stain the slides in quinacrine mustard. So quinacrine mustard is another dye which is used for staining of the chromosomes. That can be used quinacrine mustard 5 mg in 100 ml, or one can use 5% quinacrine dihydrochloride. That is an alternative dye that can be used. Either one can use quinacrine mustard or one can use quinacrine dihydrochloride for 20 minutes. When we put the slides in quinacrine mustard, there is a banding pattern appears on the chromosomes. That banding pattern is called as Q banding. Rinse in the phosphate buffer and mount slides in the same buffer. Examine the slides under fluorescent microscope. So this is the procedure that how Q banding is performed by using the dye. The name of the dye is quinacrine. So Q banding is another pattern for studying the chromosomes. In Q banding, we use quinacrine dye instead of the Giemsa that was used in Giemsa staining pattern. The banding pattern that appear with the help of this dye is also similar that we see in case of Giemsa staining.